Has a master's degree in molecular sciences and a marketing degree from the Netherlands Marketing Institute. He joined Agendia a year after it got founded in 2004 as the director of business development. Then he got promoted and became the vice president of marketing. And in 2012, you were brought here to the Irvine facility that Agendia started, and he became the VP for clinical affairs at that facility. So he hails from Irvine, and he's going to share his view on molecular diagnostics from the Agendia perspective.
So as Ahmed said, spin out of a Dutch cancer clinic. And interestingly, in the 80s, the Netherlands Cancer Institute decided to snap freeze every tumor of the patient that entered the clinic and keep it in liquid nitrogen and then in a minus 70 degrees freezer. So as a result, we have a tumor bank with about 40,000 snap frozen specimen. The other benefit of the Netherlands is that if you travel 150 miles, or what I did today from Irvine to LA, they speak either German or Belgium or French. So the mobility is not that big in Holland. And as a result, for clinicians, it's very easy to follow patients. So we have about 40,000 snap frozen specimen with more than 25 years clinical follow-up. So in oncology, that's a great resource to start developing tests upon. So you can go back to the past, and what we did in the area of breast cancer is actually say, okay, we go back to samples from between 1980 and 1990 in the area of breast cancer, and now look at patients that developed metastasis versus that did not develop metastasis. Under the microscope, they look the same, but at that time, also in the 2000s, we got a technology from this area actually, at the time developed at Rosetta in Seattle, but then later licensed by Agilent, which was the microarray technology. So for the first time, we could look at the full genome on a gene expression level. And so we took those tumors that metastasized versus those that did not metastasize, read the activity of all the 25,000 genes in the tumor genome, and said, which genes behave differently in those metastasizing tumors versus the non-metastasizing? And we found a very distinct panel of 70 genes that very accurately predict whether the breast cancer is going to metastasize or not. So that was published in Nature in 2002. In 2003, Agendia was founded to commercialize and develop this product. In 2004, and I wasn't aware of that, Bob, but our first deal was with MPI. So we went to Arizona to say, okay, we want to be present in the US market, but we don't want to do it ourselves because we were busy enough developing the test and get everything and look at the European market. But then in 2006, the FDA knocked on our door and said, well, what you do might not be fully in compliance with US law. And us being a European country took it very serious. Our competitor got a similar letter, Genomic Health, and they just sent a bunch of lawyers to the FDA saying you don't have any authority, which later turned out that they indeed didn't have. But we accepted the invitation and went for an FDA approval process. So we got a clearance in 2007 for our MammaPrint assay, and the dynamics changed completely. So we moved away from MPI, and 2008 I moved to the US for the first time to set up the commercial organization, and later on the laboratory. Time-wise, it would have made more sense to start in Boston, because then at least you have some same-day overlap with Europe. But yeah, the climate, literally and figuratively speaking, is much better here. So it was a very tough choice, and in the end we said, we'll go for Irvine. But seriously, there are a lot of labs over there. Ahmed showed already Clarient, Abbott, So there's a number of labs, so it made it a lot easier to set a facility up there, which we did in 2008. We got our CMS reimbursement for our assay in 2009, and we have had very stable reimbursement since. Also, most of the private payers actually have followed and reimbursed our assay. And that gets me to kind of the first element where I'm in slight disagreement with what Bob was saying earlier, is about, I think, but it's particular for cancer. So if we want to move to personalized medicine, most of the cancers will become an orphan disease. So I do agree with you that, low cost, high volume is one of the solutions for those areas where there is high volume. The space we're in, oncology, if we really go to personalized medicine, it's going to be a bunch of orphan diseases, and the volume will simply not be there. So the battle is going to be to capture the value. So we got 2 CLIA labs, one in Amsterdam and one in Irvine. And the other aspect I want to briefly talk about is indeed the companion diagnostic, what Martha was referring to, saying, yeah, we actually found out that the most successful way to do that is with an intermediate party, which means that we are involved in 2 very large clinical trials that are run by the academic centers, not by the pharmaceutical companies themselves. So they are investigator initiated, and it's our experience that it's much easier to do diagnostics in that setting with an intermediate factor in between, rather than to do business directly with pharma companies. So the biggest trial in the US is called the I-SPY 2 trial. It's a very interesting trial where a number of pharmaceutical companies actually test their experimental drugs very early on in the cancer treatment process, in the neoadjuvant setting, which means that the patient has not undergone surgery yet, the tumor is still there, and you already start treating. And the big benefit in oncology by doing that is that you can actually see whether a drug is working by scanning and see whether the tumor is shrinking. And if you remove the tumor and you give therapy, you'll get the answer in 5 or 10 years because the patient has developed metastasis or not. But in the neoadjuvant setting, when you leave the tumor in initially, you can actually measure whether the tumor is shrinking or not, and the FDA is very favorable to taking that as a surrogate endpoint for efficacy. That trial is completely biomarker driven. Our MammaPrint assay is the entry criteria, because the other argument is our test actually gives you a high or a low risk of distant metastasis. So why would you test experimental drugs on patients that have a low risk of distant metastasis if the drug is to prevent metastasis? So our drug is at the front end, or our test is at the front end to select patients to be enrolled in that trial, and only high-risk patients are enrolled. And then secondly, we run again the full genome chip to see if there are signatures of gene expression that can predict response to experimental therapy. It's a standing trial, which means there's no beginning, no end. It will run as long as there are drug companies that actually make their drug available, their new experimental drug available for breast cancer. So we're now testing the 4th drug in 4 years, and it's going very well. Going to the next slide to today's discussion. Again, oncology, a very specific segment, but we're spending about 90 billion on cancer therapies alone globally, so approaching 100 billion. Most of that money goes to the pharmaceutical companies. Cancer is one of the worst offenders if it comes to efficacy. At best, 25% response rates on today's drugs, which means that 75% of that money is wasted. And here I say there's a great opportunity for the diagnostic industry to actually show value, because if we can use those drugs more efficiently, the savings will be there, which is very much in the benefit of patients, because they will forego the toxicities, or they will get the right therapy at the moment in time that they need it. But again, also it will be a big impact on savings in the healthcare system in general. So I think the opportunity is there, but there are some complicating factors. So one is absolutely, what do we do with validation? We have run a prospective randomized trial with 7,000 patients in Europe to get where we are. That's a $40–50 million trial. That's not sustainable. So I agree with your remark also that we got to figure something out to do that in a different setting. we make use of registry trials a lot now. So almost all the diagnostic patients that we test also go into a registry study, so that we collect information on thousands of patients on an annual basis. But because I think you can never recoup the money spent on prospective randomized trials. The other thing is, I don't know to what extent people are familiar here with either the Myriad or the Prometheus case if it comes to patents, but that wasn't a very good thing either for the diagnostic industry, because both patents were rejected, and as a result, the patent office said that a lot of the things that we do are laws of nature, which are not patentable anymore. And you get into the situation, as Bob alluded to earlier, that if we cannot protect our diagnostic test, there is no incentive to develop clinical data and evidence to it. So it's something I don't have a solution for today. It's something that popped up relatively recently, and we just got to deal with it. And then talking about CLIA versus FDA, or laboratory developed test without FDA oversight versus FDA oversight, we clearly made a choice in 2007. Would we do it again? I'm not entirely sure, because we expected actually the FDA to step in much earlier to also keep our competitors to the same standards, which they didn't do. So we are a lot more restricted in what we can claim. We have 6 FDA clearances because our assay had its improvements over time that we had to resubmit. So it's a lot of regulatory burden. Actually, it carries a lot more weight outside the US. So we have a lot more business because of our FDA clearance outside the US than within the US, which is a bit of an awkward situation. So that's it from our end. If there's questions later, I'm more than happy to talk about it.
Thank you very much. Put your hand together for Mr. Van.